microbial growth control. So we're antimicrobials. These are drugs that inhibit the growth of microorganisms. So you can have synthetics, as it sounds, they're produced in a laboratory. Antibiotics, these are produced by bacteria and fungi. Semi-synthetics, they're chemically altered form of antibiotics. So there's only so many methods or actions, which we'll talk about, that you can kill microorganisms. And so sometimes they've gone in and kind of they took what nature had, the bacteria or the fungus, their antibiotics, and they kind of supercharged them, trying to make them stronger. So this is why you have your semi-synthetic drugs. You can have bacterial sides. So remember we talked about sides again, you see the ending, sidal, bacterial sidal, killing. So it's an agent that kills the bacteria versus bacteriostatic. So again, this is where if you learn some of those, because some people, if you really learn the vocab, learn some of the roots, learn some of the prefixes, suffixes. So you see bacteria and sidal, it's killing bacteria versus bacteriostatic, it's inhibiting, say, a static level with the bacteria. And so it's a way that you can very easily start, you don't have to memorize everything because you can figure it out or break it apart. Okay. You can use chemotherapeutics, drugs that act on diseases, very common cancer patients. So, antibiotics.
Sorry, can you go back? Can you go over the distinction between the synthetic and semi-synthetic again?
So with the synthetic, they're completely produced in a lab. So we have, it's one of those things where we're like, okay, well, we want to do this. And so a lot of times with the synthetic, they'll go through for drug production, and they make chemical compounds, and they make slight tweaks and variations. So like you remember how all those little tweaks and variations make all those phenol compounds? They'll do that, and they'll get an entire huge cluster, hundreds of these. And so they have these chemical compounds, and then they'll start screening things against it, going, ooh, that one killed it. And then they'll look in that family and go, well, maybe some of these others will do it better, or great. And so they're just chemical compounds that are completely artificially made and tweaked in a lab.
And antibiotics falls under synthetic drugs, or?
Antibiotics are usually produced, they're naturally produced by bacteria and fungus, but we produce them in a lab, but we've taken them from them. So, like penicillin and stuff, we get those from the microbe, but nowadays you don't want to harvest it necessarily, you want to be able to do it in a lab.
So, but
it's
like
not,
categorically, would it be semi-synthetic or synthetic? Like if
I wouldn't call them.
Let's say that was a question on a test.
I would, because I personally would say that synthetic means that it's in a laboratory, and antibiotics means it came from bacteria or fungus.
So would it be synthetic or considered?
I wouldn't consider it synthetic just because it came from a living organism. We aren't artificially creating a chemical compound. We're creating it in a lab. That's probably why it's put up that way, but it's kind of a natural compound.
So you would consider all antibiotics to be semi-synthetic?
Yeah.
Okay.
Or yeah, because
I think that's what he says.
because they're, I don't know. I don't know that I would consider them semi-synthetic, because semi-synthetic to me means that you took a natural antibiotic and then changed it. So I would consider antibiotics to be natural. And then the semi-synthetic means that you tweaked what nature already had, and synthetic means we played with the chemical in the lab.
Okay, thanks.
Other questions? Antibiotics. So these are chemical agents produced by certain microorganisms that either inhibit or kill other microorganisms. So some of them just inhibit growth. Some of them actually can kill other microorganisms. They have selective toxicity, so they will harm microbes without damaging the host. The whole lovely goal of it. This is this is the microbe's defense system. You know, if they can secrete some kind of antibiotic, then they can keep other microbes away from their territory and their food supply. So this is their way of carving out their territory, their food. That's why you get that zone of inhibition. It's because they're killing or inhibiting others. They also have a spectrum of activity. So you can get some, like tetracycline, which has an effect on gram-positive and gram-negative bacteria, or you can get a very narrow spectrum where it can only affect a certain type of bacteria. So there's a wide range, which is why there's so many antibiotics out there. There's, you know, only so many mechanisms of action to kill the microbe, and also what range do we need? Do we know what the patient has, so we know exactly what antibiotic to give them? Or, you know, you want to make it as narrow as possible, yet broad enough to get what you need. If you go too broad, you're killing everything. I mean, antibiotic against life. So, not something you really want to be dosing patients with really broad antibiotics for long periods of time.
Are those terms like relative, broad and narrow? Like anything that affects more than like two things, is that considered broad, or is it just
I think that's a big gray scale of, it can go from affecting almost everything to very specific, and there's a range.
Okay, so
it's just more of a,
when you're comparing two things, one would be narrow and one would be broad, something like that.
yeah, I mean, it's, where are they on that scale? Do they kill everything, or do they kill only one thing? Or do they kill 5 things? And this one kills 7, so it's more broad than the other.
When you say one thing, you're not just talking about one kind of bacteria, you're talking about one spectrum of bacteria, aren't you?
Yeah, like the coccal bacteria, one type that.
One kind,
yeah.
one particular grade, okay.
Synergism and antagonism. Okay, so if you add so this is where pharmacists have to know their stuff, because if you add 2 drugs together, and the 2 together are greater than the sum of their parts. So basically, this one, you give this one, and you give this one, and you're going to have this zone of inhibition. When you put them together, you're going to have an even bigger zone of inhibition, because they are working better together than either of them alone. Antagonistic, they interfere with each other. So, I mean, this is one of those common things. Why pharmacists have to know what's going on, and whenever you get a new prescription, pharmacist kind of checks, what are you on? And this becomes a problem for some of the older patients who have a lot more drugs, because they want to make sure everything you're taking will work either together or not really care. So you don't want anything antagonistic. Additive drug combinations, they don't really do anything, but they don't help or hurt the other one. It just, they're both there, so it's a good thing. Okay. So a pretty picture showing synergism between 2 antimicrobial agents. So you can see that each of them has their zone of inhibition, this region here. But then all of this inside here is all extra from when they just kind of, low levels of them leached out, and so they are greater than the sum of their individual parts. It looks prettier on your handout.
So I'm sorry, maybe I didn't go to sleep last night. The green area is where the 2 drugs have interacted to additionally kill more things than they would have, so this is synergism. Okay.
Okay.
Catch that the first time.
Yeah. Example of
it's up there.
synergism.
It's up there, I'm sorry.
Try to add lots of things on slides. Okay. Then we have beta-lactose. This is penicillins. This renders penicillium inactive. Okay. So this is one of those that would be more of an antagonistic, because it can actually render penicillin inactive. And what it does is it comes in, beta-lactamase comes in, and it breaks this bond, penicillin. And so you end up with an inactive form of penicillin. So, this is something you'd want to know so that you weren't giving your patient penicillin and something that has penicillinase and is going to destroy the penicillin, because otherwise, why are they taking it if you're just going to deactivate it? So, although most of us don't get penicillin anymore anyways, because more people are allergic to it, but it still is a great example of antagonistic drugs or microbials. Okay. Properties of antibacterial drugs. So, you can inhibit cell wall synthesis. You can inhibit protein synthesis, cytoplasmic membrane integrity, nucleic acid synthesis, metabolic pathways. You can also inhibit the pathogen recognizing host. DNA replication. Since it's there. So, transcription, translation. When you mess up any of the basic things, you're going to be able to kill the microbe. Okay? These are the 6 basic modes of action your book talks about. Okay? Kind of look like this. So you can inhibit cell wall synthesis. So again, this is where knowing if it's gram-positive or negative really makes a difference, because certain drugs can mess with that building of that wall. If you inhibit protein synthesis, there's multiple ways of doing that. One of the things I liked about this was it also gave you some of the drugs that actually are antibiotics that did each of these different actions. Okay, so disrupt the cytoplasm. Yeah, punch holes in the cytoplasm, it's going to start leaking. Mess with any kind of general metabolic pathway. Those are important. If you don't do metabolism, you don't really survive. Let me put it together. So if you inhibit DNA or RNA synthesis, so if you stop being able to do replication or transcription, you have issues. And then there's some newer drugs that are coming out, or they're finding out, that are actually inhibiting the pathogen's ability to recognize or attach to the host. Okay. Back to the beta-lactam drugs. So these inhibit cell wall biosynthesis. Okay? And so this is where they mess with peptoglycan cross-linking. Okay, and their big thing is that they have that, they contain a beta-lactam ring. Okay, so this is a key part in this family. This is the most common agent, most prominent in this group, the beta-lactams. They bind to enzymes that cross-link the NAM subunit. And so what happens is the bacterial walls weaken the cells. And so the cell walls start to weaken, and eventually you lyse a cell. You start cross-linking, if you start cross-linking the cell member, things inside the cell wall, then it's going to start losing its integrity. Cell walls are for a purpose. If you don't have a very strong cell wall, you're going to have problems, and you're going to break the cells. Semi-synthetic derivatives of this are more stable in acidic environments. So it's one of those things where I'm like, okay, this is great. The supplement does this. It's able to do this. Now let's take it into a lab. Let's make it more stable. Let's make it so that it's more readily absorbed. Let's make it so that it doesn't deactivate as easily, so that it actually has a longer time inside of the body. So those are all things that take a natural product and then do some of the semi-synthetic stuff to it, tweak it, and make it better.
So would the end product be considered semi-synthetic then?
Yes. So simple beta-lactams are effective only against aerobic gram-negatives. But they've made them not so simple anymore.
Wait, what only works against gram?
Ampicillin.
Super simple. Yeah.
Oh, thank you.
So,
Ampicillin.
penicillin. They're effective against gram-positive, so penicillin G. On a weird page. That's what I wrote. Okay, penicillin G. It's effective against gram-positive bacteria. Ampicillin is effective against gram-positive and some gram-negative. So you can see how starting to tweak things, you start to get the other types of inhibition. And so, I mean, if you look structurally, they're very similar. But those small tweaks can greatly improve your ability and your effectiveness. Question? We're just looking at the differences. Yeah.
What is beta-lactam?
That was what we talked about. This, they're just a class of drugs that have this beta-lactam ring. So an example, you know, this is kind of one of those things, what you can see, you try and are streaking out your Staph aureus, and then all of a sudden you have something growing, and you have this zone of inhibition. So obviously whatever that is growing produces something that inhibits bacterial growth. This is how they first started coming up with these antimicrobials, is because they were seeing things like this. And so we now know, okay, well, if we put penicillium, the fungus, on there, it's going to stop the bacteria. So this is one of those kind of like, this is how they were discovered, this is how we can still prove that they're functioning, you know? And it's one of those things when you're doing science, you streak things out, and sometimes you can get those really nasty looking things that inhibited the growth of everything around it. Do that again. Okay, so how is this working? So, I told you that it was inhibiting the cross-linking, or it's causing cross-linking. So, normal bacteria cell walls, you know, you have the NAM, the NAG, you get that 4 little tetramer cross-linking here. So these are supposed to cross-link to each other to build a nice stable wall. And that's part of what a peptidoglycan is. You know, that's part of, you know, you remember way back when we first were talking about this, the different subunits that make up the cell wall and your oligopeptides. Okay? So they're supposed to do this so you get a nice firm structure. Well, what happens is instead of cross-linking where they're supposed to, the cross-linking gets inhibited. So you get cross-link inhibited by the penicillin. And so if they're not cross-linked properly, then it's going to start being fluid, and you're going to start losing some of that stability. And so the previously formed ones can remain, but it stops the new ones from forming. And eventually, the cell will burst due to osmotic pressure because the integrity of the peptidoglycan is not maintained. Okay? So down here is some chemical structures of some of the beta-lactam drugs. Okay? So the general, you know, the mode of action is mess with the cell wall, it loses its integrity, cell bursts. Okay. You can also get antibiotic resistance. So penicillase, see ASE, an enzyme doing some kind of action. So penicillase cleaves the beta-lactam ring of penicillin. So here you have a way some of the microbes have gotten to be penicillin resistant, antibiotic resistant, because they can create this penicillase so that they can then destroy the penicillin and they can grow. So there's a constant battle between, you know, having microbial drugs and having antibiotic resistance, trying to, you know, everything's just trying to live and survive. And so, with the inhibitors, amoxicillin, so it's a semi-synthetic penicillin, or you can also have non-competitive inhibitors of penicillin. So there's multiple ways of trying to inactivate cell. Inhibitor of cell wall synthesis. So, we talked about messing with the integrity of the cell wall. You can also prevent the cell wall from actually ever being made. And so you have bactracin, a topical application that's effective against only gram-positive bacteria. So this actually will block that transport of the NAG and the NAM from the cytoplasm. Or you can have the vancomycin, which blocks the alanine-alanine bridges between the NAM subunits. And so you end up having, so this is again only against gram-positive. And then you can start putting things in there like the INH, so it inhibits synthesis of the mycoyl acid, which is used in that wall. And then this is how you start building up those triple antibiotic ointments, as you start going through and grabbing different antibiotics and kind of mixing them together, which ones are going to, so we have a broader spectrum. And this is what polysporin is multiple antibiotic units. Okay. So, you mess with the cell wall, you can mess with the cytoplasmic membrane. Here's a couple of drugs that will actually disrupt the cytoplasmic membrane. Polyvinix B binds to the cell membrane, disrupts the structure and permeability. So again, cell membranes are supposed to be semi-permeable. If you mess with that semi-permeability, then you have problems. They're supposed to be able to keep things out and keep things in. You can also get the pollines, which will bind to the sterols, the ester sterols, and the plasmid membrane. So this is great for disrupting fungal plasmid membranes. And then the azoles, which inhibit ester ergosterol synthesis in fungus. And so some of those are really nasty stuff. But you can treat some of those systemic infections and topical infections by using one of the azoles. So, where do we see these triple antibiotic ointments? You might start noticing some of the names or recognizing like, oh, okay, because you want to make sure that you're trying to get rid of most of the microbes that could be growing. So how does it disrupt the cytoplasmic membrane? One method is to actually go into the pore and create kind of this pore. And so you have your phospholipid fungal membrane. Remember, it's always a bilayer. The heads are hydrophilic or hydrophobic? And these are? Okay. And so you have these pores because you want there to be you want fluids and things to be able to go in and out of cells, but if you all of a sudden put big gaps, you can start to keep it open permanently or make it so it's not quite so rigid on what it allows in or out. And so just by shoving this into the letting it bind up the pores, it now messes with the cytoplasmic membrane, and you're able to kill cells because they can't contain what they want to contain. Okay, so those are some things you might see for when you're dealing with the antifungal medications. And I think we're all too hot and tired to really care anymore. So we'll leave inhibitors of protein synthesis. Okay, you guys, I'm just going to tell you what we're doing for the lab, so if you need to take a break, you can after we talk about it.